Greetings. What we're going to do now is shift into nervous tissue. This is very important because this is going to be the platform or the foundation from which after this we're going to be exploring areas such as the central nervous system, the peripheral nervous system, the autonomic nervous system, and then special senses. So the key to this is keep in mind nervous tissue consists of both neurons and neuroglia. And we'll get into all of this. So we're going to get started. The nervous system is divided into the following. The central nervous system, CNS, which consists of the brain and the spinal cord. The peripheral nervous system, PNS, contains both the sensory, which would be the afferent nerve pathways, and the motor, which are the efferent nerve pathways. Remember, afferent refers to heading toward a source, and efferent is heading or directed away from a source. The motor nerve pathways consist of two divisions here. The somatic nervous system, that's pretty much controlling things like skeletal muscles. The autonomic nervous system is the automated system that you don't have to think about, that you don't have voluntary control per se, but it controls a lot of the functions of various glands and smooth muscle and cardiac muscle systems. So this is, heart, digestive system, the methods to control blood pressure, etc. The autonomic nervous system is subdivided into the sympathetic nervous system and the parasympathetic nervous system, but we'll get into that in a subsequent chapter. We also have the enteric nervous system, ENS, and this is basically the nervous system that's located in the gastrointestinal wall tract and controls digestive functions. Now, as you see here, we have sensory receptors here. They follow and provide information on the sensory division of the PNS. Now, this can be both internally as well as externally. Afferent pathways direct information to the central nervous system for information processing. The information processing will pass out of the CNS into the rest of the peripheral nervous system via the motor division of the PNS. Now, the motor division of the PNS includes the somatic nervous system, SNS, and that'll be involving skeletal muscles. The autonomic nervous system, as I mentioned before, is subdivided into a parasympathetic division and a sympathetic division. The parasympathetic involves glands, cardiac muscle, smooth muscle. The sympathetic involves adipose tissue as well as glands, cardiac muscle, and smooth muscle. When we talk about neurons, these are nerve cells specialized for intercellular communication. They consist of the structure of dendrites, the cell body, which is referred to also as the soma, and the axon. Dendrites receive stimuli either from the environment or from other cells, and these can include other neurons. The cell body, the soma, this is where you have the nucleus and other organelles like mitochondria, the endoplasmic reticulum, etc., located. And then we have the axon, which may be very long or very short, but can be as long as a full meter in length away from the cell body. It carries information to other cells. Now, dendrites are highly branched. They carry fine processes called dendritic spines. In the CNS, they receive most of their info from the spines. The cell body, the soma, has cytoplasm surrounding the nucleus, and this is called the perikaryon. The cytoskeleton is composed of neurofilaments, similar to the intermediate filaments, and they can form bundles called neurofibrils that provide internal support. Here you see a diagram, and you have the perikaryon right around here, the neurofilaments you can see here, and the neurofilaments are also going to exist down in the axoplasm to help maintain the length and structure of this axon. We'll get more into that in a minute. Here you see the cell body, the soma. It consists of the nucleus, nucleolus, You're going to have a certain number of mitochondria present, as well as clusters of rough endoplasmic reticulum, which are referred to as Nissl bodies. You will also have the presence of free ribosomes making other proteins. The dendritic spines, as you can see here, 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 these are the ends of the dendrites. Then you have the axon hillock. Right just after the axon hillock is the initial segment where action potentials start and travel all the way down the length of the axon. The membrane of the axon is called the axolemma. The cytoplasmic material that's inside of the axon is called the axoplasm. Then we have these divisions. Now, some books will talk about these divisions being collaterals. Others will call them the telodendria. At the end of these are little swellings that you can see called axon terminals. This is where you're going to have synapses to communicate information from one axon to other cells. The other cells may be neurons, maybe glandular cells, muscle cells, etc. Excuse me. The axon hillock is really the origin of the axon. The initial segment next to the axon hillock initiates action potentials, as I've mentioned. I've mentioned that the axolemma is the specialized axon plasma membrane. The axoplasm is the cytoplasm of the axon. Now, it contains not only neurofibrils and neurotubules, but also lysosomes, mitochondria, and various enzymes. You have to understand that this is a pathway to send material down the length of the axon to the terminal endings, the synaptic endings. Okay? The telodendria, these are axon endings which end as a series of fine endings, and that is where you're going to have the axon terminals. They're blunt endings of the axon, and they allow for synaptic connections to other cells, and the release of the neurotransmitter occurs at this end. So here we have a telodendrion of a presynaptic cell, and so this is going to be an example, representative, an example of a synapse. Notice that you're going to have mitochondria in here, and these are important to produce ATP that's necessary for the function of this structure. You have vesicles. These are synaptic vesicles. They are loaded with neurotransmitter. You have a space here, approximately 50 nanometers wide, that is called the synaptic cleft. This is the postsynaptic membrane here. This is the cytoplasm of that postsynaptic cell. This is the presynaptic membrane. And we're going to get into the mechanisms how an action potential travels down here. But the way you have communication from one cell to the other is via the release of neurotransmitter. Now the synapse, as I've said, the space between the presynaptic and postsynaptic cell, you're going to have this exchange of neurotransmitter for communication. The presynaptic cell releases the neurotransmitter from the vesicles. That's by exocytosis. The postsynaptic cell membrane has neurotransmitter receptors. It is these receptors that are specific for the neurotransmitter, and they are involved in changing certain activities, sometimes allowing ions to flow in and out, sometimes generating small excitatory postsynaptic potentials. You'll see more of this in a bit. Now the collateral branches, these are axon branches that carry simultaneously the action potential that allow the neuron to communicate with more than one cell at the same time. The ganglion is a collection of neuron cell bodies. When we talk about a nerve, this is a bundle of axons in the PNS. When I talk about afferent fibers, these are axons that carry information to the CNS. Efferent are axons that carry information from the CNS. And you can see the synapse with another neuron here. Here's a collateral branch here. Neuron 1, the dendrites. Action potential would travel down. It would go along at the same rate of speed, etc. The same pattern by the collaterals. And you would then communicate here with neuron 2. Now, a neuromuscular junction Very similar, except the only difference really is that you have skeletal muscle fibers here. Now, you notice that the telodendria, you're basically branching off all of these structures so you can have multiple synapses on the same muscle, in this case, multiple muscle fibers at the same time. A neuroglandular synapse, again, basic difference, you have gland cells as being the receiving end of the action potential and the neurotransmitter release. Okay, so here they call this a neuroglandular synapse, here they call this a neuromuscular synapse, and here we just use the term synapse with another neuron. Now, neurons are classified on the basis of structure and function, so you need to be aware of this. Interestingly enough, we have anaxonic neurons. An beforehand is basically saying not, so you have no axon, lots of dendrites. These are found in the brain and special sense organs. Unipolar, these are dendrites and the axon are fused together. You see these mostly in sensory neurons that are unipolar. Bipolar, these are where you have dendritic processes that branch at the distal tip, the cell body between the axon and the dendrite. These are rare. They occur in special sense organs such as sight, smell, and vision. And then you have multipolar, 2 or more dendrites and a single axon, most commonly found in CNS and found in all motor neurons. Here's the axoaxonic, as you can see here. Lots of dendrites, no real clear axon here. Here, unipolar. Coming down, the cell body is sort of in the middle. You have an axon clearly defined, and then you have axon terminals. The dendrites all summate into this one single dendritic process. Now, what's the difference between that and a bipolar? Bipolar, interestingly enough, is dendrites here, and the interesting thing is the initial segment is right after the dendrites. The rest of the length is axon all the way down till you get to the axonal terminals. And you look on the side here, sort of this on the side cell body here. Now, multipolar, this is where you really see a lot more of this in microscopic slides and other things like that. You have the cell body to which has the dendrites attached right to the cell body area. Then you have the axon hillock here, and then you have the initial segment right after the axon hillock, and your axon can be very, very long. In some cases, it can be a meter or longer. Now, excuse me, let's talk about the function of this. You have sensory receptors, interoceptors, proprioceptors, exoreceptors, and then sensory ganglia. So let's clarify a lot of this stuff for you. The sensory receptors, this is found in the PNS. These are cells monitored by sensory neurons or specialized sensory neurons. Interoceptors, intero refers to inside. These monitor for digestive activity, pain, deep pressure, cardiovascular, respiratory, digestive, urinary, or reproductive organs. So these are the ones that provide you with the inside information, whether there's something wrong, whether the tissue is damaged, whether you're not getting enough oxygen, etc. Some of the information you may be aware of, some of it you may not be. That doesn't mean that it is wasted. It may be involved with the autonomic nervous system. Proprioceptors. These are structures that monitor position and movement of skeletal muscles and the joints. So you can tell what is the position of the joint. And this information will feed itself back to various structures in the brain, including the cerebellum. Exoreceptors, when we talk about exoro, it's referring to outside. So this is where you monitor external environment for heat, pain, pressure sensations, etc. The sensory ganglia is also found in the PNS. A ganglion is a collection of nerve cell bodies. Sensory from the outside world, you're going to have them in they monitor the somatosensory, and on the inside, they would be monitoring the viscerosensory. All right. When we have interneurons, though, these are you're going to find in the central nervous system, CNS. They lie between the sensory and the motor neurons, and they process sensory information into actions. So this almost goes back to that homeostatic mechanisms that you saw in Chapter 1. Sensory information comes in, and then you have the interoreceptors. They may be very simple, 1, 2 cells, or very complex. But then there is the decision of what type of response, if any, is required. And in some cases, it may be a motor response. Also, you'll find interneurons that process higher functions, learning, memory, planning. Now, somatic motor neurons are in the CNS. These innervate the skeletal muscles. The visceral motor neurons are also in the CNS, and they innervate all other non-skeletal effectors, glands, cardiac muscle, adipose tissue, etc. Now, autonomic ganglia are found in the PNS. These are divided into sympathetic and parasympathetic. Now remember, ganglia is a collection of the cell bodies. Okay? So, the set of visceral motor neurons that basically innervate peripheral effectors. Fancy words of saying, look at these motor neurons will then innervate or make connections with heart muscle, the smooth muscle in the intestine, the stomach, the blood vessels, etc. The muscles that are involved in contraction. These are all smooth muscle now. For the urinary bladder, etc. Now, neuron cell bodies are located in the peripheral autonomic ganglia. You're going to see these. Then we have somatic effectors, and the effectors are really what's being acted on. So the somatic ones you're always going to see as skeletal muscle fibers. The visceral effectors, these are the smooth muscles, the glands, the cardiac muscle, and the adipose tissue. Now, this diagram gives you some perspective here. You start with sensory receptors. If we're talking about inside the body, that's interoreceptors. If we're talking either outside of the body or in feedback or information about positions of muscles, bones, joints, that's proprioceptors as well as the exteroceptors. That would be, for example, in the skin, etc. They're going to have their afferent fibers send information up, somatic sensory neurons, visceral sensory neurons, through the PNS, then getting into the CNS. This sensory input from the visceral systems will be processed by interneurons. The somatosensory neurons will also feed back to interneurons as well as into the somatic motor neurons. And then you'll have the visceral motor neurons feeding information out for responses. These will be all coming out by efferent fibers, but they could be autonomic ganglia. They could be somatic motor neurons to skeletal muscle. And then we have the sympathetic and parasympathetic, and that's where we feed information to the various other visceral effectors, smooth muscles, glands, cardiac muscle, adipose tissue. Now, we got to talk about also the neuroglia of the CNS. Now, I hope you still have that concept map for the nervous tissue and the different types of cells. That will help you in reviewing these principles. Let's talk about some of these cells. We have the ependymal cells. These form simple cuboidal or columnar epithelium that line the cerebral spinal fluid-filled areas of the brain and spinal cord. For example, the ventricles, the lateral, the third ventricle, the fourth ventricle, etc. The central canal that you find in the spinal cord. Now, ependymal cells assist in producing, monitoring, and circulating CSF. So where you have the choroid plexus, which is this little tuft of capillaries, it's going to be also covered then by ependymal cells that will control what is being secreted out of these capillaries, and then as it passes through the ependymal cells, this is what will be finally made as the resultant CSF, cerebral spinal fluid. Microglia, developmentally, they're related to the monocytes and macrophages. These are a type of immune cells. They migrate into the CNS during formation, and yet they persist as guard cells in the CNS for the rest of your life. And they basically, their job is to remove waste, remove cellular debris, and pathogens by phagocytosis. Astrocytes, they help maintain the blood-brain barrier. They isolate the central nervous system from blood, such as the blood's chemicals, the hormones, etc. They regulate basically gas, nutrient, and waste, in other words, other substances, in the interstitial fluids surrounding the neurons. They will form scar tissue after CNS injury. Now, we're still in the CNS, so it's important to keep this in mind. Oligodendrocytes provide the structural framework within the CNS by stabilizing the position of the axons. It is the oligodendrocytes that also will do the myelination. Okay? The myelin sheath, which is this wrapping of cell membrane around a length of axon, will enhance the speed of action potentials, APs. So what you have basically is that instead of action potentials traveling along the entire length of the axon, that entire length of axon, all you have are gaps where the myelin is absent. These gaps are referred to as the nodes of Ranvier. So an action potential will jump from node to node to node, which means that the action potential will travel much faster along the same length of axon when you compare that to an unmyelinated axon. Now, there's certain areas where you're going to have predominantly lots and lots of myelinated axons, and this is referred to as white matter in the CNS. The areas that are referred to as gray matter in the CNS are either made up of neuroglial processes. It's basically either going to be cell bodies or axons, but that are not covered by myelin or neuroglia. Right? Sorry about that. Here is that concept map that I mentioned to you before, and you get yourself 1, 2, 3, 4, 5 questions, and you can figure out what you've got. Does it produce action potentials? Yes, so it's a neuron. Does the cell form a myelin covering around the sheath? Yes. Is it present in the central nervous system? Yes, then it's an oligodendrocyte. If it's no, it's a SCHWANN cell or a satellite cell. If it does not have myelin covering around the neurons, does it wrap around the brain capillaries? If it's yes, then it's an astrocyte. If it's no, then the last question is, does the cell act as an immune cell? That would be the microglial cells if it's yes. If it's no, it's ependymal cells. Here is a diagram that you can see here. And this is taken from basically an edge between the gray matter that is on the inside of the spinal cord and the white matter that is on the outer edge of it. Now, central canal is here. So you have the ependymal cells right here. You have a neuron. Now, the neuron cell bodies are going to have other cells connect with them. For example, you're going to have, in this case, this is an astrocyte. But you're also going to find some cells in there. One example would be the microglia. But notice that this is primarily gray matter, and that's because there's very little myelination. You're going to get much more myelination, so it'll appear somewhat whitish. When you have a lot more myelinated axons present, these will be provided for, at least in the central nervous system, that includes the brain and spinal cord, with oligodendrocytes. And if you notice, they extend their cell membrane and wrap around a bunch of times tightly around these axons. Now, one more thing, astrocytes. They extend what they call pseudopodia, which are extensions of the cell membrane, to wrap very tightly around and basically cover the entire capillary. You will see this extensively in the brain. The blood-brain barrier, and I'll probably reiterate this later on, is exceedingly important because you really not only control what substances pass from the blood into the brain tissue, but you protect whatever is in the blood from damage by these substances that could go into the brain tissue but are blocked because they're either charged or they're too large of a molecule, etc. Paul Ehrlich, an early physiologist who did remarkable work, did a simple experiment. He injected trypan blue, which is a staining dye, into an artery in an animal. And he noticed that all of the tissues in the body, except the brain, took in this dye. So he thought there's got to be some sort of barrier, some sort of protection for the brain. But then what he did was he injected the same trypan blue into one of the ventricles of the brain, and he found that all of the brain was stained with this dye. None of the other tissues were. And that further reinforced his theory that there was this. And after a while, with proper cytology and proper tissue analysis, we've come to find out that the blood-brain barrier is very important. It will block certain toxins from going from the blood into the brain. We'll get more into this later on. Here's the ependymocyte right here. They have cilia here. They will also have basal processes, and they will have connections over to the astrocyte. Here we have the oligodendrocyte. The area that it myelinates and covers the axon is referred to as the internode. The space in between is the nodes of Ranvier, so that's where you're going to have an action potential jump from here to there, etc. When we look at the cells, we notice that this is a neuron here, and it has lots of synapsing with other cells, other related cells. Here we have an unmyelinated axon, and the reason you say it's unmyelinated, even though there's some astrocyte attachments, is that you have a complete exposure of the length of the membrane. Okay? Then we're going to get into some of the neuroglia of the PNS, the peripheral nervous system. Now, Schwann cells, also known as neurolemmocytes, cover the peripheral axons in two different ways, as well as participate in axon repair after injury. Satellite bodies are another type of neuroglia of the PNS, but not everyone is really sure what they do. They do know they surround the peripheral cell bodies. Now, of course, myelinated cells, as I mentioned here, the outer surface of the Schwann cell, is called the neurolemma. The Schwann cell will protect the myelinated and unmyelinated axons from contact with interstitial fluid. Unmyelinated cells, though, have Schwann cells that stabilize the position of the axons in clusters. That's where you're going to start getting the formation of, we would call these nerve fibers. They're really made up of a collection of nerve axons. The Schwann cell plasma membrane wraps around the axon like a jelly roll, wrapping layer upon layer upon layer, and it squeezes out a lot of its own cytoplasm. So all you have is really just the lipid cytoplasm as this layered approach wrapping around the axon. Okay? Now, each internode you need to be aware of is composed of just a single Schwann cell. And you can see that right here. Here's her cell body. Here's the nucleus. Here's the dendrite. This is the axon hillock. This is the initial segment where you're going to start off action potentials. Here you have an internode. Here is a node of Ranvier. Each individual Schwann cell is here. If you'll notice, it is wrapped many times around the axon. And so you really only get a lot of notable cytoplasm around the outer perimeter, as well as the nucleus here. Okay? You have the axolemma Here you have the axon here. So the axolemma is the cell membrane here. Now, compare that to a peripheral a PNS cell that has, it's not myelinated really. What have you got? Well, notice that you've got some satellite cells kind of clustering around the soma of this axon. You got a variety of axons traveling kind of parallel, but they have some exposure to the outside. You have a SCHWANN cell that basically acts to kind of wrap around the axons, but not completely. And what it does is basically stabilize the position of these axons altogether. Okay? This is a process that you can see here of how you literally will have the SCHWANN cell surround a portion of the axon within the groove of its cytoplasm. So you see the SCHWANN cell here, and it's going to continue to wrap and wrap around, and as it does, a lot of the cytoplasm gets squeezed out and forced to the leading edge of the SCHWANN cell. So the SCHWANN cell will always at the end have sort of a leading edge where you have more cytoplasm, where you have the nucleus, but all of these layers are tightly wrapped around, and this forms what we call the myelin. The next area I want to do is bring up to you axonal injury and repair. And as I mentioned earlier, the SCHWANN cells play a very important part in this, but this is only really seen in peripheral nervous system nerves. The person that finally gets it all figured out and gets it done so that you can do regeneration repair of axons in the central nervous system, particularly in the spinal cord, probably will get the Nobel Prize. Okay, whether it's a team or not, it's going to be a Nobel Prize event. Why? Because everybody is trying to seek out means to treat and repair spinal cord injuries. And if that happens, then you're going to have a lot more people not needing a wheelchair, but being able to be mobile again. Now, let's just deal with PNS right now. We're going to talk about Wallerian regeneration. This is the process of SCHWANN cells repairing the severed axons, which basically is consisting of 4 steps. First, distal from the injury site. In other words, that part of the axon that is furthest away from the soma and is on the other end of the injury cut will degenerate, break down. SCHWANN cells do not degenerate per se. They will proliferate along the pathway of the original axon, and macrophages will remove the cellular debris. Now the neuron will recover, and the axon will begin out of sprouts from the proximal site and grow along the SCHWANN cell pathway. So the SCHWANN cell is sort of this basically pathway or roadway that allows to direct the axon sprouts to head down and make connection where it previously was fully connected. Now, as the axon grows, the SCHWANN cells begin wrapping around this newly developing axon to develop the new myelination. If sprouted, the axon reestablishes the synaptic connection and normal function resumes. So let's go here. Step 1. You have the injury, and what immediately happens here at the proximal stump. Now, the difference between proximal and distal is what? Distal is further away from the target, which is the soma. Proximal is much closer. So when you have this, let's say, severing of this nerve, all of this that's downstream from the cut, the injury, will basically turn into blobs. It'll degenerate, fragment up. The cut end here will begin to become a blunt end, seal up very quickly. You have the myelin here, you have the proximal stump, and you have the distal stump. What dies is that cellular material that's from the axon. What does not die is the Schwann cells. They're going to basically provide a pathway for the resprouting of the new axon. But you got a lot of cell debris there, and that has to be broken up and removed. That's where you have the macrophages. And basically, you're going to start having this proliferation of Schwann cells. They will make a pathway. Then what you have is the axon growing into the injury site along the Schwann cell path. And then eventually what happens is the Schwann cells are wrapping around the elongating axon, and eventually you have full control, full reestablishment right back, and it's synapse occurring at the next cell that it was supposed to be connected to. Now this is where we're going to start getting into the concepts that you really need to get down. So I'm going to emphasize this and encourage you to check on the videos as well as the readings on this, because neuronal activity depends on changes in the membrane potential. You can also see the module 9.5 on page 322. What you have to keep in mind is you have unequal distribution of positive and negative charges across the membrane surface. This is going to create a potential difference, or what we call a membrane potential. Keep in mind that the concentration of potassium ions are +1 in charge. They are going to be in abundance inside of the cell and very rarely outside of the cell. Whereas outside of the cell, outside of the axon, whatever, is going to be a high concentration of sodium ions, which are also +1. And there's very little sodium on the inside of the cell membrane. Now, this is a result of the membrane's selective permeability and active transport mechanisms. One of the active transport mechanisms that's key is the sodium-potassium pump. That is something that's going to require ATP, and it's going to basically take the sodium that's on the inside, kick it outside, and it's going to take the potassium, which is outside, and kick it back in. Now, you need to overview and review the role of membrane potential in neural activity. We need to go over resting potential, graded potentials, action potentials, synaptic activity, as well as the information processing. This is going to be connecting the stimuli to a specific cellular response. So let's get started here. First off, as I mentioned to you before, you're going to find that there is a mix of positive and negative charges on the outside as well as on the inside. But the predominance of negative charges on the inside gives the membrane potential more negative here compared to the positive out here. Mostly sodium out here. There's a few little ions that are negative, and they might be chloride. There's going to be a lot more negative ions in here, particularly because of the proteins with charged amino acids that make up these proteins. The proteins can't go either side, can't get transported through the plasma membrane. So they're going to have a particular influence in making this inside or cytoplasmic side more negative than what's on the extracellular fluid, which is outside of the plasma membrane. Now what happens is this, we're going to see this progression here. Resting potential, you're going to have a bunch of graded potentials created by synaptic connections from other cells to this particular cell membrane here, this neuron. Stimulus will produce the graded potentials. Sufficient graded potentials may generate an action potential, which would travel all the way down here on the axon. And eventually you get down to here, and that's where you're going to have synaptic activity, where the action potentials will eventually cause the fusion of the neurotransmitter vesicles to be released across the synaptic cleft, that small space there I was talking about. And that will basically lead to a depolarization of this postsynaptic cell. This is where we get the information processing. But before I continue with that, let's go step by step here. Now, first off, differences in electrochemical gradients determine the resting membrane potential. This resting membrane potential is when a membrane is undisturbed. But you have to keep in mind that you have both active and passive processes that do exist. The passive ones, you're going to find that there are potassium and sodium leak channels in the plasma membrane. These channels leak ions from high concentration to low concentration. The cytosol of the cell has many negatively charged proteins. The extracellular fluid has few. The proteins can't escape. So this is important for electrical force on the mobile ions of sodium and potassium. Then we have also the sodium-potassium pump. Now think about this from another perspective. If you've ever been on a boat, you have what they call a bilge pump. That's basically where liquid, water or seawater, will seep in. If you can think of the hull of the ship as a membrane, you're going to have a little bit of water leaking in. So you're always going to have this pump, which of course uses energy, pumping out this little bit of leakage. Okay? So sodium and potassium leak channels will allow some of this passage of ions going in the directions from high concentration to low. But you've got this pump, which is going to take the sodium and potassium, put them into the areas back in the high concentration. Potassium will be pulled from the outside back in. Sodium will be pulled from the inside back out. But it requires energy, and that's in the form of ATP. And this is going to help maintain what we call a stable resting potential. Keep in mind, we're pumping with 3 sodium ions outward, 2 potassium ions inward. Here's the plasma channel. Now look, one of the other things you need to keep in mind is that leak channels, or basically all ion channels, are specific for specific ions. Potassium cannot go through a sodium channel. Sodium cannot go through a potassium channel. They're all set by different sizes as well as charges along the length of the channel that control what ions can pass through. In this case, the plasma channel, the sodium leak ones will allow sodium to go in. The potassium ones will go out. When we take a resting membrane potential here and we put it inside the cytosol, we have a resting membrane potential here of -70 millivolts. Remember, milli is the prefix that basically says 1/1000th. So 70 1/1000ths of a volt. Okay, here's our potassium leak channel, leaks out some of the potassium, and this gets brought back inside by the sodium-potassium exchange pump, which uses ATP broken down as energy to run this pump. Sodium leak channels have sodium leak in, and then they are also taken out by the sodium-potassium leakage channel. Okay. Now, this is where we get into the concept of a chemical gradient. Now, for any ion, this is a concentration gradient across the plasma membrane. So when I talk about this right off the bat, I've got more sodiums on the outside than on the inside. I've got a sodium chemical gradient. For the potassium that's on the inside in great quantity versus outside, I have a potassium chemical gradient. But these are not just basic atoms, they're ions, so they're charged. So the electrical gradient is created by the attraction between oppositely charged ions and repulsion of the same charge ions. This is where we fuse these together and we have the electrochemical gradient. This is a combination of chemical and charge gradients and is really a form of potential energy. You see, think about this. I've got a large amount of sodium on the outside, a large amount of potassium on the inside. I have got room to draw sodium inside. I've got a pull to move potassium outside. But I've also got this membrane potential, which is the result of this chemical and electrical gradient. Okay. no net movement of ions across the membrane occurs when you have this balanced out. And that includes the balancing effect of the sodium-potassium pump. Now, the equilibrium's potential indicates an ion's contribution to the resting membrane potential. And you see this right here. Okay? Potassium chemical gradient, very great in this direction. Potassium chemical gradient, somewhat to the opposite. Potassium electrochemical gradient is really the sum of these two. Same thing here when you have the sodium chemical gradient, sodium electrical gradient, and yet you have a sodium electrochemical gradient, which is very forceful going inward. Now, there are three types of gated ion channels that change the permeability of the plasma membrane. Remember, charged ions cannot pass through the lipid bilayer of the plasma membrane. Just can't happen. You've got to have these channels here. And this is where we get into chemically gated ion channels, voltage gated ion channels, and mechanically gated ion channels. Now, chemically, otherwise known as ligand gated, means that when the ligand, the specific chemical, binds to the channel, it will cause the channel to open up. Voltage gated ion channels are a little different. They open or close upon changes in the membrane potential. Finally, mechanically gated ion channels. These open in response to the mechanical stimuli that physically distorts the neuron membrane surface. So it's almost as if you're sitting there going, hey, I'm going to flex this membrane, and as I do, the ion channels, which are mostly protein, will flex, and they will eventually be popped open or popped closed, depending. Now, the distribution of the gated ion channels on a neuron, in the soma and the axon hillock, they are chemically gated. In the axon initial segment and the axon length, they're voltage gated. And they're voltage gated for sodium and potassium channels. Finally, the axon terminals. These have voltage gated channels, but they are specific for another ion, calcium. Be mindful of the fact that calcium has a +2 charge. Okay, so they are very different from the sodium or potassium channels. Here we have an example of ligand gated. Remember, ligand is any term. So I could be sitting there saying hormone, I could be saying drug, I could be saying, let's see, neurotransmitter. In this case, we're going to use acetylcholine, which is a neurotransmitter. It binds here, causes this protein to deform, pop open. So the gated channel is gone from closed to open, and you can now have the sodium ions flood in. The channel, after the ligand pops off from this binding site, will close. Keep in mind something. There are 3 states for a sodium ion channel: closed, open, inactivated. Now you might wonder, what do you mean by inactivated? Because of this inactivated state, sort of like a rest state before it has to reset, this provides us with certain situations of threshold, which we'll get into shortly. So how many times does it take for me to stimulate the neuron to generate an actual potential? Well, there has to be a small spacing of time between them. Here, the gate is closed. There is an inactivation gate right here and an activation gate right here. Sodium can't go in. When I have a change, a depolarization that occurs, the channel opens up. Boom, sodium comes in. What happens when I get a full, complete depolarization and I'm up, notice now, plus 30 millivolts, not minus 60. I've gone all the way up to plus 30. There is a little inactivation gate that closes here. This is still open, but no ions can pass through. Now, With mechanically gated ion channels, this is a little bit different, okay? What happens here is if I apply a pressure here, this distorts the membrane. And so it causes the proteins here, which are embedded into that membrane, to flex open, and then you have sodium moving in. When the pressure is removed, the channel closes, okay? Now, as we mentioned earlier, here's the chemically gated ion channels, which will all be out here at the soma and the dendrites. You will have the axon hillock. The axon initial segment and the entire length of the axon is composed of voltage-gated sodium and potassium channels. So they're all voltage-gated. Finally, we have voltage-gated at the very tips of the terminals here, voltage-gated calcium channels. Now, graded potentials, also known as local potentials. They are localized changes in the membrane potential. They're not the big action potentials, but they can help to create the conditions to stimulate an action potential. So you want to be aware of that. But graded potentials are any stimulus that opens the gated ion channel will produce a graded potential. This form of depolarization cannot spread far from the site of stimulation. So what's the process? Step 1. The membrane is at resting potential. Okay. Step 2. The membrane exposed to the ligand that opens the chemically gated sodium channel. Fine. And as a result, step 3. Sodium ions rush in. A depolarization occurs. But here's the point. The depolarization is a change in the potential from resting potential to a smaller negative value, as well as the potentials above 0 millivolts. So you get a small rushing and you get a small localized area of depolarization. The extent of depolarization spreads and a local current forms. This is the sodium ions that follow the open channels. The degree of depolarization, though, this is part 5, declines with distance from the stimulation site. Impact of the membrane potential is proportional to the size of the stimulus. So, great amount of stimulus, you're going to get a greater amount of membrane potential. Impact, very light amount of stimulus, much lesser amount of membrane depolarization. So the greater the degree of stimulus, the greater the degree of depolarization. Okay, that makes sense. And you can see this all repeated here. There is a couple of points, though, that you need to be mindful of. Here is our resting potential. It's at the axon hillock. Here is a depolarization that has occurred, so sodium rushes in. You've gone from minus 70 millivolts to minus 65. And then what happens is this, then you end up forming this kind of local current. So it starts acting on short areas nearby, and you have from minus 70 to minus 65 to minus 60. There's a problem. It's almost as if you think about it, if you've ever done this, take a pebble or a stone and throw it into a large body of water, like a pond or a lake. You notice that the waves start spreading outward from the site where you had the original stone hit the water. The waves right next to the impact were greatly disturbed. There's a higher amount of those waves. As they spread out, though, the waves get less and less in height. So think about this. At the point of stimulation, you're going to have the greatest amount of membrane potential change. But as you spread away from that part of the stimulus, the depolarization gets less and less and eventually dies out. Now, we got to deal with something that's very important, and that's called repolarization. That's the process of returning to a resting potential of the membrane. For example, minus 60 millivolts can easily be shifted back to minus 70 millivolts. How? Sodium-potassium pump. You can also have another unusual phenomenon, which we call hyperpolarization. This is a change in the membrane polarization negatively beyond the resting potential. Now, you need to get both these terms down now because they're going to be instrumental in helping you really understand the entire setup for an action potential. Hyperpolarization is hyper, I'm increasing the polarization. So I'm going from minus 70 and I'm going to decrease it further to minus 75. So here you have on this chart, you can see minus 80 here, minus 70, minus 60, and this is a period of time. So if I have the chemical stimulus opens the ion channels, I end up with a bit of a depolarization. Once I get to sort of the top of this wave, the excess sodium ions are starting to be transported out of the cytosol, and this is also the fact that the stimulus has been removed. If the stimulus continues there, you're going to continue to keep having this depolarization occurring and may eventually get even higher. But once you remove it, then you're going to have the reversal. You're going to have a repolarization occurring, and you'll see this repolarizing back to the resting potential. Now, there are some mechanisms that allow for what we call hyperpolarization. If I have a chemical stimulus that opens the potassium ion channels, they're going to cause more of these positively charged potassium ions to rush out, which will cause, from the resting potential, the membrane potential will go lower. So it will be going down, 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 down to almost minus 75. Notice that once the chemical stimulus again is removed, it will reverse to eventually the resting membrane potential at minus 70. Now I've got you set up to understand further the concepts of action potentials, APs. Action potentials are all or nothing. One size fits all. Sorry, there's no super big ones nor tiny ones. You either have it or you do not. And it's only in one size. Prime means of communication for neurons. That's it. Begins with a membrane potential reversal. It is propagated, these are propagated changes in the membrane potential, and they travel along the axon membrane. Now, depolarization to threshold will trigger the action potential to start. It must occur at the axon initial segment. So we're going to get into this. We have to have a certain amount of depolarization that rides into or is right at the initial segment to trigger the action potential. So let's go through the steps. It's a little different. Why? Because you're going to have two sets of gated channels popping open and popping closed. First off, resting potential for the membrane, minus 70. Yeah, we know that, okay? The depolarization to the threshold, get up to minus 60 millivolts. Once we get to that threshold, we have a different set of gated channels called voltage gated channels open up. These are for sodium in the beginning. The sodium voltage gated channels open up, you have immediately rapid depolarization occurring. So the membrane depolarization will start jumping upwards to almost the positive zone. And this is all occurring very, very rapidly. Now at a plus 30 millivolt, the sodium gated channels will close. Blunk, 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 blunk. And they're inactive, so you can't reactivate them immediately. But the potassium voltage gated channels open at this repolarization or this depolarized point. And so they're going to start this repolarization process where you're basically going to start having the membrane polarization go from plus 30 to plus 20 to plus 10 to 0 to minus 10, minus 20 millivolts continuous. Once you're near the threshold, the sodium voltage gated channels change from inactive to closed. So in other words, once you move to close, you're able to reactivate them, to open them up again. This is once you're near threshold. Remember, threshold is -60. Now, the reason I say that is because as the potassium voltage-gated channels are opening up and they're starting this repolarization, once you have repolarization, or the membrane get down to about -60, then you can re-stimulate it and fire off another action potential. Now, at the threshold of -60, the potassium voltage-gated channels are closed. There's going to be some overlap in time, because once they close, you still have some that are closing, you still have some that close, but you're still going to have some of the last potassium ions going outside. And so you're going to have this hyperpolarization that occurs. So it's going to dip down a little further, and that's about -75, and then the sodium-potassium pump is going to kick in. And once that kicks in, it's going to reestablish or restabilize the membrane potential to -70 millivolts. Keep in mind, resting potential, graded depolarization, rapid depolarization, repolarization. So let's start. Your resting membrane potential is -70 millivolts. Here's a sodium, they're closed, but they could be activated. Potassium, closed. So you go depolarization to threshold. So you have this local currents, graded potentials that cause us to move from -70 to -60. And boom, now you activate the sodium ion channels that are voltage gated, and they're all going to pop open, and suddenly you're going to have this inrushing of sodium ions. They're positively charged. So you're going to have the membrane potential go from -60 to +10 to eventually +30 millivolts. Once you hit the +30, the sodium ion channels, the voltage-gated ion channels, close. Blunk. But at the same time, you're going to have the potassium ion channels open up. And now you're having this outward rushing of potassium ions, +1 charge. So you're going to start reversing the membrane potential till it gets lower and lower. Now you might have eventually some hyperpolarization. In this diagram, they say -90. A lot of books will say -75. It doesn't matter. You have that overshoot of hyperpolarization, and then it's going to be restabilized by the sodium-potassium pump, and you have the -70 millivolts. Now, remember I talked about the inactive phase of the sodium voltage-gated channels? Well, this is where it contributes to what we call the refractory periods. There's two types, the absolute and the relative. Now, the absolute refractory period, this is a situation where the membrane cannot respond to further stimulation. There are no action potentials that can be generated at this point. The relative refractory period, this is a membrane can respond to stimulus, but it must be much greater than the original stimulus, a larger than normal stimulus. So let's take a look at this from a action potential. What gated channels are opened up, and which ones close, and which ones contribute to the absolute refractory period, and which ones contribute to the relative refractory period. So we get a depolarization, localized, graded depolarization. You get up to your threshold right here, -60. Boom. Sodium voltage-gated channels pop open, and you have this rapid depolarization which rushes upward and shuts down at the +30 millivolts. This is a point where the sodium channels close. Blink. But they close in an inactive state, meaning no matter what amount of pressure, no matter what amount of stimulus you give, they're not going to reopen again, and so you're not going to be able to generate a second action potential yet. as you hit this point, plus 30, what also is going to occur as the sodium voltage-gated channels close, the potassium voltage-gated channels open. Now you're going through repolarization. Potassiums are going out of the cell. And you're moving through the absolute refractory period till you get down to about here, which is minus 40. Some books would say it would be down here, but whatever. It's down here. Guess what happens? The sodium voltage-gated channels that were inactive now get flipped into closed, which means that they could be activated. The only problem is that you need a much greater stimulus to get them to fire off again and have another action potential generated. Once you get down to here, minus 60, the voltage-gated potassium channels begin closing. You'll still have, and all of this is not like turning a switch on and off. It's similar, but you're going to have some lag time within this period of time. And as a result, you're going to still have some potassium leave, and so you're going to end up with an overshoot here. Your resting membrane potential is minus 70. This may shoot down to about, minus 75. But at the point you are here, all the sodium voltage-gated channels, all the potassium voltage-gated channels are closed. So how do you get from minus 75 back to minus 70? Sodium-potassium pump. The sodium ions get kicked back outside, the potassium ions get kicked back inside, and you have a resting membrane potential. Now, action potentials may affect adjacent portions of the plasma membrane through continuous propagation or saltatory propagation. We'll get into saltatory in a second. When I talk about propagation, it's the same repeated process of the action potential traveling along the axon membrane, okay? Which is an interesting point, because how is it that it always knows how to go in one direction only along that entire length of axon? That is, how does the action potential just go in one way? Well, remember, the parts of the axon that have just had an action potential go through have got a pause now because of the absolute refractory period. So they're only going to go in the direction where they can still be activated, etc. This is continuous propagation. The propagation of an action potential along an unmyelinated axon or muscle plasma membrane, in which the action potential affects every portion of the membrane surface. So every part that is not in absolute refractory period can be stimulated, can move along in this depolarization, and continue to have an action potential travel down it. What about saltatory propagation, Saltaire leaping? This is where we got into what we talked about before about myelinated axons and the nodes of Ranvier. So the relatively rapid propagation of an action potential between successive nodes of a myelinated axon is really saltatory propagation. Instead of going through all the entire length of that axon, you're only going through just small segments. The ions cross the plasma membrane only at unmyelinated nodes. This is the nodes of Ranvier. And when the action potential starts, they skip the internode, because really in the internode there's no sodium-potassium channels underneath. And they depolarize the closest node in the process. So you can see here the action potential, initial segment, this is segment 2, segment 3, and what happens is this. Right now you get into this and you're in a threshold, so you can't go but in one direction. Okay? Now, saltatory propagation, though, you got one other little thing you got to keep in mind. You've got your internode here, which is myelinated. So you have this action potential develops at the initial segment, and then you have the ions jump over to here, where you have another plasma membrane that is exposed, and you go through the changes here. and you go through the depolarization, as you can see, and then you have it jump over to the next node. So basically, when you look at action potential propagation, there are two means to do it. One, myelination. Two, increase the axon diameter. The larger the diameter, the lower the resistance to ion movement, and the faster the action potential travels. Let me give you a little sidebar here. There's a wonderful place where National Institute of Health scientists go for the summer. It's called Woods Hole. Yep, Cape Cod. And they used to go out every day and have a ship go out to pick up squid and other aquatic animals for marine biological experiments and for general biology experiments. One of the interesting things that they went for squid was because squid have a very large diameter axon. And so what they do is, as soon as they bring the squid in, they dissect out that axon and they study it. They study the membrane, they study the axonal plasm inside, and they did a lot of these experiments so they could understand how an action potential travels. And so the two basic differences, or two means to speed up action potential, are the action potential propagation, I should say, is the presence or absence of myelination. The more myelination you have, the faster the action potential. The greater the diameter of the axon, the faster the action potential. Otherwise, if you have a really, really small in diameter axon, it'll be much slower than if you had a big, wide, large diameter axon, okay? If you have the loss of myelination, we're getting into that in a second, you will have action potentials that will eventually be generated, but they will be much slower from going along the length of the axon, okay? Now, we're going to get into this a little further later, but let me move into the other part, which was information traveling. Keep in mind something. Information in the form of action potentials travels throughout the cell to the next cell, but the information that is being communicated from one neuron, let's say, to the other, or from a neuron to a muscle cell or a neuron to a gland cell, you have to jump across the synapse. And there are two types of synapses, chemical and electrical. Most of us in our body have chemical. There is a few unique cases of electrical still, but most of them are all chemical, okay? And when you look at action potentials, the maximum amount, if you looked at the chart, you saw that it takes about 2 milliseconds to complete an action potential. Now, if you remember that milli is 1/1000th of a second, and there are 1,000 milliseconds in 1 second. That means that ultimately the maximum amount in 1 second of action potentials that you can generate is 500. Now, if there are no variations in the action potential, in other words, no king-size ones, no tiny ones, then how do you communicate information? You put it into a code, and the coding is basically the number of action potentials per second. The maximum amount that you can generate per second is 500. But you can generate a small amount, a medium amount, or a large amount. And that's how you're going to be able to pass information of, let's say, intensity or volume or whatever else you need to transmit. Let's get back to synapses, though. Chemical synapses rely on chemical release from the presynaptic neuron terminal membrane, and the chemical opens ion channels on the postsynaptic membrane. This is the most common synapse. Electrical rely on the presynaptic and postsynaptic membranes locked together by gap junctions. The action potential is transferred from membrane to membrane. These are rare. They are found a little bit in the brain, eye, and ciliary ganglia, but they are also found somewhat in developing embryos. Now, the synapses that use acetylcholine as a neurotransmitter are called cholinergic. Okay. So here we have an example of basically a chemical synapse. You have your presynaptic neuron. The action potential comes down to the end here. You have extracellular fluid here. You have your synaptic vesicles here. You have mitochondria here. You have also a very small space here. This space is your synapse. It's about 50 nanometers. Nano is billionths of a meter. And you also have an enzyme that will destroy the neurotransmitter. It's called acetylcholinesterase. Okay? Now, there's one other thing that you have to keep in mind. As the action potential comes down here, you have voltage-gated, wait for it, calcium channels, calcium ion channels. What happens is that as the action potential travels down here, it pops open the calcium ion channels, calcium streams in, which causes the vesicles containing the acetylcholinesterase to migrate to and exocytotically fuse with the terminal membrane here, which will cause them to dump their acetylcholine, the neurotransmitter, out into the synaptic cleft. Now, keep in mind that these little purple structures are the chemically gated sodium channels. So what happens? Once you have this release across, they're going to travel very quickly across this synapse. You have then the initiation of an action potential. How? Because once the neurotransmitter, acetylcholine, binds onto these chemically gated sodium channels, they pop open. Sodium rushes in. This creates a graded depolarization, which will eventually, if it hits threshold, achieve the initiation of an action potential down the length of this membrane. Now, that's turning the switch on. How do we turn the switch off? In other words, how do we reverse all of this? Well, you don't have to reverse it in the sense of pull up all of the neurotransmitter. There are some neurons that do that with other neurotransmitters. But in this case, what we do is we have an enzyme sitting right here in the synapse that will break down the acetylcholine. And that enzyme is called acetylcholinesterase. It is an enzyme that will break down all of these acetylcholine molecules. You will get acetyl group and you'll get choline. Choline will eventually be reuptake into the basically this presynaptic terminal, and it will be remanufactured into more acetylcholine, and the acetyl group will kind of float away. And you can see this diagrammed here in the steps. Action potential comes down, calcium ion channels that are voltage gated. So the change in the polarization right here, because of the action potential, will cause these gated channels to pop open. Sodium rushes in. As sodium rushes in, you now have the synaptic vesicles fuse with the terminal membrane. They will release acetylcholine. It will bind to the acetylcholine-gated sodium ion channel, allow sodium to rush in. You have depolarization that's beginning. To deactivate this entire signal, you have an enzyme called acetylcholinesterase, breaks up the choline from the acetate. The acetate floats away. The choline is reuptaken and fused to make more acetylcholine neurotransmitter. Now, this is the other example here where you have an electrical synapse, and that's basically you have the presynaptic neuron. The action potential travels down here. It goes through and starts depolarization here of the postsynaptic neuron, and it's basically because you've got gap junctions here that connect presynaptic with the postsynaptic neurons. Now, Postsynaptic potentials are responsible for information processing in a neuron. Postsynaptic potentials, these are graded potentials that develop in the postsynaptic membrane in response to the neurotransmitter binding to the receptor. You want to keep in mind any single neuron may have hundreds of synapse connections at the same time. In other words, you have lots of information coming into this particular neuron. Some of it is going to be causing depolarization. Some will cause hyperpolarization. So let's talk about these postsynaptic potentials. There's excitatory and inhibitory. Excitatory postsynaptic potentials, EPSP, these are graded depolarizations that are caused by the neurotransmitter that shifts the membrane potential closer to the threshold. Makes sense. But we also have IPSPs, which are inhibitory postsynaptic potentials. These graded depolarizations are caused by the neurotransmitter that shifts the membrane potential farther away from the threshold. So instead of going from -70 to -60 with the EPSP, the IPSP might cause it to go from -70 to -75. And it's also interesting that when you get a stimulation of an EPSP and an IPSP at about the same time at the same place, they can literally cancel each other out. This is where we get into concepts of summation. This is the integration of the effects of graded potentials on a segment of the plasma membrane. Two forms can trigger an action potential. They can occur in time or in space. In time, we call this temporal summation. And they can occur when a single synapse is stimulated repeatedly at the same time. The summation of the graded potentials allow for the membrane depolarization to achieve threshold. So you keep pounding away at this depolarization. You go from -70, you're -65. At the same time, you're hitting it again, -60. And then you're at threshold and boom, then you start an action potential. But you could also do this in space, on the neuron membrane. That is what we're talking about. This is called spatial summation, involving multiple synapses that are active at the same time. And the depolarization effects are cumulative to achieve the threshold. So with the temporal one, they all have to occur at the same time. The spatial, they don't have to occur at the same time, but they can occur by multiple synapses acting on the membrane at the same time. And so what happens is you get this cumulative effect. All right. Here's an example if we look at this. Now, this would be, of course, time, and this would be, of course, membrane potential, -60, -70, -80. I have a depolarization stimulus applied right here. And so as a result, as it continues, I have an excitatory postsynaptic potential. This is called facilitation. Okay? So I'm getting closer and closer, but it's continuing. When the stimulus is removed, this graded potential breaks down very quickly. Now I have resting potential. Now what I'm going to do is hyperpolarizing. So I'm going to have the hyperpolarizing stimulus applied, and now I have inhibited, because I have the inhibitory postsynaptic potential. When the stimulus is removed, very quickly, the hyperpolarization returns back to resting potential. Now, what happens if I had both hyperpolarizing and depolarizing stimulus applied at the same time? You would have the summation of an EPSP and an IPSP at the same time, and it would end up, they would cancel each other out. Okay? Now, this is more realistic of what you see with neurons. Every once in a while, you get a model, and you got one or two of these terminal buttons attached, and that's it. And people are kind of going, oh, okay. But with a lot of neurons, you don't just have one or two or maybe 10. You're getting input from a variety of other neurons. And so this is going to have a summation effect either time-wise or spatially, to cause this initial segment to start sending out action potentials. And some of these attachments will also be glial cells. They won't necessarily cause a stimulus, but they are there to help manufacture and control the amount of ions that are present right around the membrane. Here's the example of temporal summation. I get the initial segment right here. I get my first stimulus. And I have the second stimulus occurring, and so it is able to create a greater amount of depolarization at the same time, and the thresholds reached, action potential propagation starts down here. Spatial requires about the same time. You have basically several nearby synapses, and between them they create such a summated depolarization that part of that threshold is achieved right here, and boom, that leads you to an action potential propagation. Now, I mentioned this before about coding, and so I might as well bring it back again. Because what happens is a lot of my students think biology, 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 but you've been dealing with the information age, with technology, with electrons traveling around. You have computers that code information and send it off in little packets. And in a way, you can have the similar models here. When you have information processing, this involves interacting groups of neurons and various neurotransmitters that encode information as action potential frequency. When I say frequency, let's say number of action potentials. Since they don't change in size or length of time, you have one AP, it's the same as every other AP. So the way that you convey information from that action potential is that you change the amount per second. The maximum amount that you can have per second is 500. The coding of information is based on the number of action potentials per second, as well as the neurotransmitters from various neurons. To help you to think about this, remember we talked about EPSPs and IPSPs? You have to understand that some neurotransmitters will trigger and will bind on to ligand-gated channels that create a hyperpolarization. Others that bind to a sodium or potassium ion channel, and they will cause a depolarization. So you have to keep in mind that when you saw that image earlier with all those synapses attached onto the surface of the soma of a neuron, you've got negative influence, positive influence, all occurring at the same time. So it's sort of like, well, all the pluses and all the minuses, what do you get in that particular area? Now we have regulatory neurons. These are neurons that regulate information by facilitating or inhibiting activities of the presynaptic neurons affecting the cell body membrane, or by altering the sensitivity of the axon terminals. What I'm saying here is you have a lot of different ways to interpret all sorts of different sensory inputs. Let's just take pain for an example. There are neurotransmitters of substance P. That is a neurotransmitter that is sent by pain neurons. But the interesting thing is that you can block its release by opiate neurons. Yes, they do issue, but not heroin and not morphine. They issue what we call endogenous opiates. These are neurotransmitters that act very similar, such as endorphins, enkephalins, dynorphin. Okay? And these will block or inhibit substance P neurons from releasing their neurotransmitter and basically blocking the sensation of pain. Here's an example of a regulatory neuron here and here, presynaptic neuron here, and a postsynaptic neuron. The presynaptic may interrupt the function of this presynaptic neuron here, or this regulatory neuron may act on it, or this regulatory neuron. Notice that its synapse is right here, right near the terminal synapse membrane. And it can release neurotransmitters right here to block the APs from this presynaptic neuron sending out its neurotransmitter to the postsynaptic neuron. And this does exist in some modes in the spinal cord. Now we're getting into neurotransmitters. There's well over 100 different ones. Some of them have direct effects on ion channels, and that's one means to affect the postsynaptic target cell. Others have what they call indirect effects that require secondary messengers. Now, secondary messengers are necessary to have the neurotransmitter affect the cell. To help you understand what secondary messengers are, let's talk about what the first messenger is first. Neurotransmitter membrane receptor is activated by the neurotransmitter binding to the receptor will bind to, and also the receptor will bind to a G protein. This is located inside of the cell. The G protein becomes activated, and now you have a G protein that will trigger a diverse family of enzyme complexes and other biochemical activity. The secondary messenger is the activated G proteins. They will trigger the release of ions or molecules. And so really you have the first messenger that is really the neurotransmitter, but since it can't go inside the cell, it binds to a receptor. The receptor has a nearby G protein that becomes activated. The secondary messenger is the activated G proteins, and they trigger a cascade of various other biochemical activities, ion releases, molecule movements, etc. And they alter the cell activity or the gene activity or biochemistry of the postsynaptic cell. You want to see the neurotransmitters and the mechanism of action of the messenger systems as well. So here's the example here. Here is the neurotransmitter epinephrine. This is a beta adrenergic receptor. This is the receptor here. Epinephrine cannot get through that membrane. Epinephrine in this case does not bind to an ion channel. But when it binds to this beta adrenergic receptor, this G protein gets activated. And it gets activated because you have GTP, that's similar to ATP, loses a phosphate group and becomes GDP. And the phosphate group phosphorylates. This G protein becomes active. It activates a particular enzyme called adenyl cyclase. The adenyl cyclase converts ATP to cyclic AMP. Cyclic AMP will trigger a whole host of other activities. Now, how do we reverse cyclic AMP, shut the switch off? Because turning it on is important, but also understanding how it turns off. There's an interesting enzyme called phosphodiesterase here. And phosphodiesterase breaks away the cyclic structure of the molecule, and so it becomes just plain old AMP. Now, gee, that seems kind of complex, but it's not. Once you understand, there are a lot of drugs that act on secondary messenger systems. I've got one that you probably take, well, if you're a teetotaler or if you're a coffee drinker. It's called caffeine. What caffeine will do is it will block phosphodiesterase. When it blocks that, that means that your cyclic AMP, which is also called the secondary messenger system. This is the first messenger. This is the second. I'm trying to clarify and correct what I said earlier. This cyclic AMP will stay longer and activate biochemistry of this cell. Everybody got that? Longer, because phosphodiesterase has been inactivated because of the presence of caffeine. That's why you get part of the perk that you sense. Okay. Now you'll see this in your book. And first off, no, you do not have to memorize all the chemical structures. Okay. Don't lose sleep over that. Next, a general understanding of the mechanism of action is only necessary. I would encourage you to review the location of where some of these are and some of the comments. The key ones you're going to learn a lot more of, both in the early part of the nervous system and in the autonomic nervous system, etc. And then later on, we'll probably go into some more of it in ANP2, is acetylcholine. Those are known as the cholinergic neurons that have receptors for acetylcholine. Norepinephrine and its we'll call it sister or clone, epinephrine. You'll notice that the chain is very similar to each other. They only just add on this methyl group here on the end. This you'll see, it is indirect. It requires G proteins, secondary messengers. You will also see a lot of times norepinephrine and epinephrine not merely as a neurotransmitter, but as a hormone. Serotonin, you'll see this a lot. As a matter of fact, we use certain selective serotonin reuptake inhibitors, SSRIs. Prozac is one example. And what these drugs do is to enhance mood and to act as antidepressants and things like that, anti-anxiety. These drugs will block the reuptake of serotonin, so it stays longer in the synapse and continues to enhance certain mental states of the individual, particularly by enhancing the hypothalamus and the limbic system. Then we have glutamate and GABA, which is gamma-aminobutyric acid. Notice that GABA has both direct and indirect systems, whereas glutamate has both direct and indirect. When we talk about direct, it might be an ion channel, etc. Indirect, that's where you're going to have the G proteins and secondary messengers. So I would encourage you to review this information processing section. This will be very helpful for your understanding of both presynaptic and postsynaptic responses. Well, it looks like we've completed this. That's great. What I encourage you to do for this chapter is to peruse chapter review outline on pages 431, 432. To complete the review questions on pages 433 and 434. To complete the review sheets on pages 412 and 430. Now, the next chapter we're going to deal with is the spinal cord. It is extremely important that you get through this chapter and make sure that you have a good handling of it before we get into the spinal cord. Also, don't forget to review the videos that are for this particular lecture and prep your reading and do that before you start up with the lecture for chapter 12, the spinal cord. And I hope you have a great day.